So our next presenter today, we're very grateful to have Dr. Gerhard Wagner with us today, the professor at Harvard Medical School. Dr. Gerhard Wagner is the Elkan Blout Professor in the Department of Biological Chemistry and Molecular Pharmacology at Harvard Medical School. Wagner obtained his PhD from the ETH Zurich in 1977 with NMR studies of protein dynamics. Subsequently, he worked on methods for protein resonance assignment and structure determination. From 1987 to 1990, he was a faculty at the University of Michigan in Ann Arbor, where he developed triple resonance methods for protein assignments and dynamics. Since 1990, he is professor of biological chemistry and molecular pharmacology at Harvard Medical School. His current interests are on eukaryotic translation initiation, transcriptional activation, membrane proteins in micelles and nanodiscs, and also GPCR signaling mechanisms. A major effort is on discovery and characterization of small molecule inhibitors of protein-protein interactions. He is also strongly interested in further development of NMR methods and procedures for advanced sampling and data reconstruction. Dr. Wagner received the Laukian Prize of the ENC, and he has been elected to the German National Academy of Sciences, the National Academy of Science USA, and the American Academy of Arts and Sciences. Today, his talk is entitled The Power of Isotope Labeling NMR of Proteins. Without further ado, Dr. Wagner, you can go ahead and share your screen, and the floor is yours.
Thank you, Kelly. Thanks for inviting me to talk to us. I have to warn you, my internet connection is rather unstable and it may drop out at some point. Can you see my screen?
Yes, we can.
Okay, so since this chem's isotope is in isotopes, I want to talk mainly about the benefits of isotope labeling for protein for protein NMR. So just to start, it's a long way from the first NMR spectra of ribonuclease. This was from Saunders, Fisher, and Kirkwood in 1957. I recorded my first 2D spectra of a protein, BPGI, in 1982. And this was all before isotope labeling was possible. And with isotope labeling, you can get very nice spectra, and the dramatic step in the information you can get from proteins with isotope labeling. So I determined my first structure of BPGI based on homonuclear NOEs before isotope labeling, and the spectra just got the right fold of the protein, but we're far below the quality or resolution of the crystal structure. And this was all before isotope labeling. With isotope labeling, we can get very nice structures. Here's just an example which I want to discuss today. It's a structure of the MET25 VP16 complex based on N15 carbon 13 isotope labeling. You see the MET15 structure is cis, and the bound activator is rather flexible and binds only at certain points, and many parts are unstructured and flopping around, very mobile. And for this, to essentially get this structure, it was very important to use some special isotope labeling. So we could only be folded correctly by identifying NOEs to one aromatic side chain, which sits up in here. So we are very much interested in aromatic side chains labeling, with aromatic side chains form major parts of protein cores and protein complexes, but are underutilized in protein NMR. So we get help from smart isotope labeling and from aromatic CH trosis, like first published by the Baruchin lab. And we can also get some information about mobility of aromatic side chains in the interior of proteins. So this is again the system. It's a complex between the MET25, a part of the mediator complex, which interacts with a transcriptional activator domain from VP16. And since the system is also used for yeast-2 hybrid labeling binding experiments. So we are interested in interaction of this. And the aromatic side chains play an important role. And this is now a spectrum of uniformly labeled carbon-13 labeled HSQC spectrum of this MET25 co-activator. You see the spectrum is very unresolved, almost unusable for more detailed structural analysis. And then we went into this area of alternate carbon-13, carbon-12 labeling, which was introduced originally by David Lemaster in the Richards lab, by many others. And several of my postdocs and students worked on this system, starting from Cole Takeuchi, Alex Milburn, Harry Arsenari, and many, many more. And this is a scheme where you can use different precursors, 1, 2, or 3 carbon labeling of pyruvate or glycerols, and then you get aromatic side chains that have different carbon labels in there depending on what precursors you use. And we use this. And here is a simulation of spectra of BClXL with the different carbon-proton correlations in this region of the proton-carbon COSY spectra. And if we do uniform, and these are experimental spectra with BClXL, if you use uniform carbon-13 label, this aromatic spectrum is almost useless. But if you use 2 carbon-13 pyruvate, then you get one set of signals for tryptophan and phenylalanine. If you use 3 carbon-13 pyruvate, you get another set of aromatic cross peaks, and then it becomes much more useful for structural analysis. And here is a spectrum from this MET25 EP16 complex. This is uniformly carbon-13 labeled, and if we do it 2 carbon-13 labeled and do a TROSY, you get a much better spectrum, and this becomes usable now. Okay, and so we tried out how BClXL looks like if we use uniform labeling, 2 carbon-13 pyruvate and 3 carbon-13 pyruvate. You get different subsets of the signals, and if you do it in the TROSY experiment, you can go through 1 times circling and 3 times circling around constant time circling, and you get different spectra. You get a high resolution if you just go one constant time region. But if you run it 3 times a constant time region, you get very sharp signals, and this can be quite useful. And so we also use this TROSY together with NOSY. Aromatic to methyl NOEs can be observed with an aromatic TROSY NOSY, but cannot be seen with uniform carbon-13 labeling. In this publication, we showed this is this pulse sequence for this aromatic NOSY, TROSY experiments. And if you compare here, if you do this experiment, you can see nice cross peaks between the aromatic side chains and methyl side chains with 2 carbon-13 pyruvate. If you just use uniformly labeled carbon-13, none of these cross peaks can be seen. You gain a lot if you use this TROSY and the 2 carbon or 3 carbon-13 labeling. And with this system, we could get a structure of this complex. You see here the MET25 in the cartoon representation. And in order to get this structure, we had to analyze cross peaks between a tyrosine in this pocket here with some other methyls or aromatic residues from the mediator. And here we can see clearly intermolecular NOEs between MET25, phenylalanine, and alanines from the ligand. So we can see intermolecular NOEs only with this 2 carbon-13 of labeling of the protein. So we can set intermolecular NOEs and teratomic interaromatic NOEs in this pocket here. And this was crucial for getting the structure solved. Otherwise, we couldn't have solved it. And here you see a cartoon of it. In blue is MET25. In yellow are parts of this VP16 transactivator, and only part of them are structured, and two different parts interact with MET25, with the mediator, and connected by flexible loops. And before we could only see where it binds, but using this aromatic mediator contacts with a special labeling, we could get the whole complex determined. So here it was quite important to have this alternative labeling available. Now, what about fluorine-19 labeled aromatic? So it was mentioned before. We were intrigued by the high sensitivity of fluorine-19 and started to explore what could be done. It's known that fluorine has a large chemical shift anisotropy and is cause for Trosi experiments. But in order to use it, you need a dipolar partner. So you looked for carbon-13 fluorine-19 pairs. And there should be no one-bond carbon-13 couplings. Otherwise, the spectrum would be very difficult to analyze because it would be very complicated. And so we got into synthesizing specifically labeled amino acids, and this was done with the help of Cambridge Isotope, where we got the initial form, and then we worked with Vlado Gelev, a previous postdoc of my lab. He's now faculty in Bulgaria at the University of Sofia, and he can put the fluorine on there. This is his characteristic picture, and he helped us making this fluorine labeled and tyrosines and phenylalanines with precursors we obtained from Cambridge Isotope. And to incorporate synthesized aromatic amino acids, we use glyphosate inhibitors, G-chemic acid pathway, and this inhibits all normal synthesis of aromatic residues in expressed proteins, and so we can add fluorine-labeled amino acids, and they get incorporated in the proteins. And then it was important to calculate the Trosi effect for best fluorine-19 location. And we used the spinach program from Ilya Kuprov, and he was collaborator on this project. And we could make this and make some proteins labeled and test what works. And this is Ilya Kuprov, now in Southampton. And he can synthesize the Trosi and the quality intensity of the peaks for Trosi and decoupled signals. So blue are the decoupled signals, and this is dependent on the field strength. So we were wondering what spectrometer frequency would be best. And this is for carbon-13 in dipole contact with fluorine. The decoupled sequences, pulse sequences gives this intensity. So Trosi peak gives a minimum around 600. If we do carbon-13 protonated Trosis, so this is transverse relaxation rate. So we want to be at low values to have the best observation. So carbon-13 bound to protein gives a much higher, larger relaxation rate. So we want to be here. If we do a fluorine coupled to carbon, this is all gets very bad at high magnetic field, and also proton coupled to carbon, Trosi and decoupled Trosi is very bad. So this is clearly gives the shortest transverse relaxation rate. So this is the way to go. So, and here is an example of a Trosi effect observed in a carbon-13 spectrum of a GB1 protein at multiple magnetic field strengths. So this is just this fluorinated tyrosine from 400 megahertz to 900 megahertz. You get the highest sensitivity at 900, but it turns out that 600 probably is the best. So it doesn't look so, but here you get the sharpest lines. Also you see this is a spectrum from GB1 from a 3D experiments, and you see the Trosi peak and the anti-Trosi peak. So we want to go for these Trosi peaks. And then the Trosi effect, the carbon-13 fluorine Trosi effect depends very little on the molecular size in terms of correlation times. This is for different correlation times. If we use this carbon-13 fluorine Trosi effect, it's almost independent of some molecular weight. So this could be something used for very large system in solution NMR. And this is the best carbon-13 detection of fluorine coupling. This is carbon-13 bound to proton. So this is not as good, and proton Trosi changes a lot with molecular weight, and also fluorine detection is not very good. So the best is to do carbon detection in a fluorine dipolar complex. And here are similar simulations for different aromatic side chains, 2 fluorine phenylalanine, 4 fluorine phenylalanine, and so on. So these and 3 fluorine fluorophenylalanine would be good. So here are a couple, this one and this one are the best. And 3 fluorotyrosine would also be good. So we have just tried the tyrosine so far, which give very nice results. And if you compare carbon detection of carbon fluorine pairs with fluorine detection of carbon fluorine pairs, the fluorine detection is certainly not as good, which comes out from the simulations from Ilya Kuprov, but carbon detection of these pairs is the way to go, and you can get very sharp signals in these systems. And then we can do a Trosi experiment. We can, for example, do fluorine start experiment where you start with fluorine and detect fluorine. Then this is a spectrum from I think it's GB1. It's not so good if we do start with carbon-13 and detect fluorine. This has the highest sensitivity, but it has not so sharp lines. The best is to go for fluorine starting and detect carbon-13. So this gives the sharpest lines. In this case, we expect 15,000 peaks and more, and there are more. Some may be flipping slowly. So this is maybe an indication of some dynamics. So the best thing is to start with fluorine and detect carbon-13. And this is because the relaxation time, the transverse relaxation time of carbon-13 the shortest, so you get quickest recovery. Fluorine has this is the longest, so you get the best signals because the longest T1 values. Fluorine has the shortest T1 values, so you can use this for rapid recovery. So it's good to start with fluorine and detect carbon-13. And these are spectra of a system that has this number of fluorines. This is, we get the best spectra because we detect carbon-13 and we recycle with the fluorine. And this is has shortest relaxation times. We give the sharp has the shortest relaxation times, so you don't get the nice signals. And you see that there are some broad lines which may be due to some aromatic mobility. And so we can also see mobility. If you in maltose binding protein has 17 tyrosines, and we can make them fluorine labeled, and I get relatively nice spectra if you get TROSY for carbon detection, but much nicer if you do the fluorine detection of the fluorine-carbon TROSY. And you see almost the right number of cross peaks, much better than with the carbon detection. And even if you go down to 10 degrees, the lines completely broaden out with carbon detection, and you get very nice spectra still at that low temperature with fluorine, with carbon detected fluorine-carbon TROSY. So what about nucleotides? Harald talked about this. Ilya Kuprov simulated this, and we see the TROSY of different nucleotides. And again, this is the in blue is a decoupled spectra, and in red are the TROSY components. And we see the best. We have obtained uracil from in collaboration with Quacudai and Christoph Kreutz in Austria, and this gives a rather nice low transverse relaxation rates. The best would be fluoroadenine, but it's difficult to make, so we have not got any of these. And this could be very nice for ATP, using fluorine-carbon TROSY for ATP, which comes, of course, in many biological systems. So, and we had some collaboration with Christoph Kreutz, who made us a tar head loop, and we get very nice uracil TROSY peaks, and Quacudai transcribed us some RNA, and which has this shape, and we get very nice peaks in here. So we can apply this for small RNAs, but we have not really proceeded on this in some biological experiments. Final thing to say is, if you have a fluorine-labeled residue like tyrosine in a protein, like here it's GB1, and then you can essentially saturate the tyrosine, and then you transfer strong NOEs to nearby residues, and you can read out with an N15 HSQC of this protein where you saturate one fluorine, and you can, if you know the structure, what's nearby, from the peaks that show up in this different spectrum, you can identify this tyrosine to what residue belongs. Or if you irradiate another one, like this tyrosine 33, you get another subset. You get a picture what is around this tyrosine. And this could be quite interesting if you, for example, we are interested in GPCR, where we can ligate receptor tails outside so the transmembrane region, and could do this to find out what is nearby. If you can get selectively one fluorine-labeled residue in a protein, you can map the environment in a new system, such as interface of GPCRs on the edge of the membrane. And here's another case.
Gerhard, I apologize for interrupting, but we have exceeded our time limit. If you would be able to, okay, you have one more slide.
Okay. Thank you. And these are the
Thank you.
collaborators on this. These are mainly those who work on the NMR. And with this, I close and thank you for listening.
Wonderful. Thank you so much, Gerhard, for this wonderful presentation. Unfortunately, because we are over the time limit, we won't be able to answer the questions that we do have in the Q&A box, but we will go ahead and we'll post those into the chat. And if you would be able to answer them after the fact, that would be greatly appreciated. And if you could please stop sharing your screen, I can go ahead and introduce our next speaker. You should be able to stop sharing at the very top of your screen. There we go. Awesome. Thank you once again, Gerhard.